polysaccharides, even phospholipids, and the lipid A is the endotoxin. Endotoxin is released when gram-negative bacteria die, and gram-negative bacteria stain red from the counter stain, and gram-negative bacteria produce non-specific symptoms. Fever, inflammation, malaise, lethargy. They spike a fever because your body doesn't see anything to fight. All it can do is raise your body temperature and try to break down the endotoxin and kill off some more bacteria. Alrighty. Family Enterobacteriaceae, gram-negative, all spiral shaped, so spirillums and spirochetes and helicobacters, those are all gram-negative. Anything curved or spirally is gram-negative. And Pseudomonas and Neisseria gonorrhea are gram-negative. Your gram-positive bacteria, they have a homogeneous wall, which means it's all the same throughout. It's 6 to 8 layers of peptidoglycan with lipoteichoic acids and teichoic acids in there. And gram-positive bacteria stain purple from the primary stain. They retain that stain throughout the whole process. And gram-positive bacteria excrete exotoxins while they're alive. So if you get food intoxication, it's from the toxins in the food, not the bacteria themselves necessarily. And because it's a proteinaceous excreted waste, your immune system will see it as a foreign protein, and you will get a specific immune response. If you get a specific immune response, you can probably create a vaccine of some kind. There's different kinds of vaccines. That actually comes up in the immunology chapter, but read it in your handouts package. That'll be the easiest way to go. So toward the back of your handouts package, you'll see passive immunization and active immunization. If it's an active immunization, what does that mean? I'm going to activate your immune system. I'm going to cause your immune system to see something, an attenuated virus or an attenuated bacterium. And your immune system is going to see it, and it can't cause disease, but it still looks like what it is. So your immune system recognizes it and learns to fight it and remembers it. So that the next time it sees it, it immediately recognizes it and starts to fight it. That's active immunization or active vaccination. Passive vaccination is when you give ready-made antibodies. So maybe the antibodies were made by a horse or a rabbit, and I give you a vaccine of these ready-made antibodies. So those antibodies from the horse go to work to recognize and fight the infection. Your immune system can take a nap. Your immune system doesn't have to be bothered because the ready-made antibodies from the horse are doing it for you. So your immune system is passive. Somebody else is doing the job for you. With passive immunization, it's short term. It'll get rid of the pathogen, but then your immune system is going to wake up and see that there's a foreign antibody there and destroy it. So it's a short term, but it's good. If you get a toxin like botulism, or if you get venom from a snake, if you take an antitoxin or an antivenom, usually what those will do is go in and work for you. And they work rapidly, because if you get botulism or a snake bite, you can't wait 2 weeks for your immune system to boot up. You need some antibodies in there right away. So they give you ready-made antibodies. And those snake serums, those snake antivenoms, from what I understand, they have to be pretty fresh. So they kind of have, there's a guy named, if he's still alive, his name was Bill Haas or something. He was from Florida. And he kept snakes of all different kinds just for that. If someone was bitten by a snake, he'd have to make some antivenom.
He's still alive.
Yeah, is he still alive, do you know? Yeah, he must be pretty damn old, and he's very old now, but yeah, I'm sure they've got people running it after he dies, but. Yep, if you get bit by a snake, you're gonna need some help quickly. Although those snake handlers, they usually try to desensitize their immune system to it by giving them little bits of venom, so their immune system builds up strength against it. Which would sort of be like activating their immune system. Alrighty, most cocci are gram-positive. Anything with coccus in the name is gram-positive. Okie dokie. And what's the STD? That's a flagellated protozoan that's going to be on every test? Good. And remember, temperature requirements of bacteria. Some are psychrophiles. What do they love?
Love cold.
Cold. What's a mesophile love?
Body temperature.
Yeah, body temperature. What's a neutrophile love? Neutral pH. Nutrients. They love that too. What if it's a halophile?
Salt.
Salt. What if it's a barophile?
Pressure.
Pressure. Likes a certain pressure. Good. What if it's a capnophile? All right, like carbon dioxide, CO2. Cap is carbon dioxide. Capnophile. Okie dokie. About that one. Okay, what kind of organism causes polio?
Virus.
Good. What kind of organism causes chickenpox?
Virus.
Good. What kind of organism causes measles? Virus. Good. What kind of organism causes typhoid fever?
Bacteria.
Good. What kind of organism causes typhus?
Bacteria.
Bacteria also, but it could be transmitted by a head lice. Good. So don't mix up your vectors with your causative organisms. Okay? And okie dokie. That one. Okay. Let's talk about different types of enzyme inhibition. Competitive enzyme inhibitors. They're drugs like antibiotics, like your classic bacteriostatic antibiotics typically. Competitive enzyme inhibitors like penicillin and sulfa drugs, they're going to compete for active sites on an enzyme that bacteria need to run their metabolism. So they can also be called anti-metabolites. Sulfa drugs is considered an anti-metabolite. So competitive enzyme inhibition means they're going to compete for active sites on a bacterial enzyme, so the bacterial enzyme cannot work efficiently. And in that way, you're going to slow down your bacterium. So it's not automatically going to go in and kill your bacteria, it's just going to harm them and make them stop growing. So it's bacteriostatic effect. The problem with that is bacteria, if they can not be killed by it, they can actually be made stronger. They become resistant. So bacteria can become resistant from competitive enzyme inhibitors. But competitive enzyme inhibitors, magic bullets, they're better magic bullets because they tend to focus on bacterial enzymes and not our enzymes. So we can actually create a chemical like penicillin, even synthetically, that will compete with a bacterial enzyme, but won't harm our enzyme. So they're better magic bullets, less side effects. Then you have your non-competitive enzyme inhibitors like heavy metals, like silver nitrate was put in babies' eyes when they were born so they wouldn't get gonorrhea from coming through the birth canal. And also copper sulfate and arsenic and mercury and all kinds of weird heavy metals. And also halogens I lumped into that group, which is not scientifically technically correct, but we can lump them together. Chlorine, bromine, bromine, chlorine, fluorine, and iodine. All of those things can damage bacterial enzymes. But because they kind of bind a different way and bind to an allosteric site and change the shape of the enzyme by breaking bonds, they can do that to our enzymes too. So non-competitive enzyme inhibitors damage enzymes, and our enzymes could also take a hit. So those can be toxic to the host. But if you go in and you damage bacterial enzymes and you kill the bacteria, they aren't going to be able to develop resistance. So each one has its upside and its downside, I guess. Alrighty. How do bacteria develop resistance to antibiotics? Okay, so how did so the patient's the one who caused the problem by not finishing their antibiotic. But how does the bacteria develop resistance when you don't finish taking your antibiotic?
Well, the antibiotic kills the weaker ones, so the stronger ones will just start growing.
And what makes the weaker ones weak and the stronger ones strong?
Biofilm.
Their genetic makeup. So what makes bacteria develop resistance is that the bacteria literally evolve. So some mutations might make some bacteria stronger and others weaker against a certain antibiotic. And also, if there's genetic recombination, that also creates new genetic types. But for bacteria, they don't have genetic recombination when they replicate. When do they do genetic recombination?
Well,
Right, when they conjugate, when there's transduction, which involves a virus, or when there's transformation, which involves competent bacteria picking up a piece of DNA from a dead cell. Good. So they evolve through mutation, and genetic recombination creates new genetic types. Then natural selection acts on them. In a way, it's kind of not really natural selection.